DNA sequences and enzymes used in the example are from a Nobel Prize winning experiment that is described in the CRISPR exercise available on the CASEIT website. The link is in the video description. My purpose here is to demonstrate features of the CASEIT simulation, not to explain the biological rationale for this experiment. That rationale is described in detail in the answer key, which is available to instructors who download the software. We'll begin the demonstration using a series of steps that are written down at the CASEIT website under CRISPR exercise part 4. I had previously opened some files and wanted to show the procedure for clearing the screen so you can start over. You simply use the clear menu and clear everything. Now that clearing procedure doesn't change the agarose concentration. The default is 1%, which is used for most of the cases. But for this particular experiment, an agarose concentration of 2% is appropriate. So to change that, you go to the DNA menu, down to Select Agarose, and 2%. And whatever the current concentration is will show in this window. Use the DNA button on the silver button bar and locate the folder that's called CRISPR data. Make sure that you've got it set on the list view. Then I'll double click on the CRISPR data folder. For part 4 of the CRISPR exercise, the sequences are in the folder called figure S15, which is one of the figures in the Nobel Prize winning research article. So I'll double click on that, and the 2 DNA files that we need are indicated here. I'll click the first, hold down the shift key, and click the second. I could just double click now, or I can click the open button. And those 2 files are going to show up in the open in process window. The last file opened is the active file, but you can change that by clicking lines in the open in process window. This exercise uses plasmid DNA as indicated by the message. Although depicted as strings of characters, characters in this particular file represent a circular plasmid sequence. In reality, the last character in the sequence would adjoin the first character in the sequence, forming a continuous loop, as designated by 3 dots at the beginning and end of the sequence. And then it says to click the sequence button to see it. So here is the sequence button. The reason why this is appearing in the lower field is because the blue arrow points downwards. If you click the white arrow, it will turn blue and the sequence would have displayed in the upper field. There are 3 periods that end the sequence and 3 periods at the beginning of the sequence. If those had not been present, then this string of characters would be considered to be a linear string rather than a circular loop. Resize the open in process window to make it larger, since some of the file names that will be generated are fairly long. Click the enzyme button and select the 5 enzymes that say Cas9-GFP 1 through 5. So I shift clicked those and I'll click the open button, and now those appear as lines in the open in process window. Next step then is to select the non-complementary strand of DNA. We're going to digest it with the first 3 enzymes, so I will shift click to activate those. Go over to the Cut DNA button on the silver button bar and make sure you go down to multiple enzymes in separate digestions. By doing that, we end up with 3 new lines generated that show the DNA sequence digested by enzyme 1, 2, and 3 for those Cas9-GFP enzymes. Select the complementary DNA sequence and then shift-click to select enzymes 4 and 5. Go to the Cut DNA button with multiple enzymes in separate digestions. Go back to the enzyme button, click that, and open up the enzyme file sal1. Shift-click to highlight the 5 digested files. Go back to the Cut DNA button and say with a single enzyme. Now activate the non-complementary DNA sequence. Go back to the Cut DNA menu and again say with a single enzyme. Click the DNA button and open the DNA file 100 base pair marker. So to recap what we've done, here are the 2 original DNA files. Here are the 5 Cas9 GFP enzymes. And we first digested the non-complementary sequence with enzymes 1, 2, and 3, right here. And then we digested the complementary sequence with enzymes 4 and 5. So those digestions are here. The enzyme sal1 was opened, and then the 5 previously digested sequences were digested with sal1, resulting in these 5 double digestions. The non-complementary DNA was activated and also digested with sal1, and the result is here. And then finally we opened up a 100 base pair DNA marker. The last step of the procedure is to load and run the gel. The fast way to do that is to click the first double digested line and then go down to the marker, shift click to highlight all 7 of these. Then go to the bottom of the Open in Process window, click the Quick Load Run button, DNA gel, load wells and run gel in one step, and stain DNA fragments purple. So that procedure loaded the 7 wells in sequence, and to see what's in them, a new window has popped up called the loaded window. And I'll move that into position. So that shows what is in each of the 7 lanes of the gel. You are looking at one of the more important results in the history of biological research. As I said at the beginning, the purpose of this tutorial is to demonstrate some of the features of the case-it simulation. It's not to explain the biological rationale of each step that we went through. For that, there is information at the case-it website, parts 1 through 4 of the CRISPR exercise. Instructors who download the software will get an answer key that gives a detailed rationale for each of these steps, plus answers to questions that are posed to students as they go through that exercise. Gels can also be run on the lab bench using simulated equipment. Click the lab bench button at the bottom of the data screen and click the toggle switch on the power source. Notice that the gel has run for 60 minutes at 125 volts. If we stain that and compare it to the other gel that we ran at 100 volts, some of those fragments ran off the gel. So we will use this controller on the power source, and you can repeatedly click that, go back to 100 volts, reload, and rerun the gel. We'll leave it stained even though that's not the way you would do it in a real laboratory, just to see the fragments move. So here is the DNA ladder at the base in the same order of fragments that we saw on the data screen. There's a timer setting. Double click on that, type in a new value, use the set button, set the time, reload it, and you can either run it realistically, or you can reload it and run it instantly as a timed run. Or you can run it as an hour run. That's the same gel that we had originally. You can also change the agarose concentration. On the lab bench, you use the run menu to do that. So there are a variety of ways that you can simulate laboratory equipment on this particular screen. Hide the gel. For example, here's a micropipetter. You could add tips to it. Dispose of tips. You can load the gel from scratch using the Tools menu, but I will not demonstrate those features here. Back to the data screen. And do an hour run of the gel, and we are back to where we started on the data screen.